Okay, we're back. Good. Right.
Well, he's still coughing a touch. So yeah.
Hi. Recording there in the front, Matt.
Happy New Year to you. Is the idea for me to present off this?
Gotcha. So then you click on it, it'll stop recording.
I don't mind. But I'm gonna have to switch screens at some point.
Yeah. Okay. So where does the slideshow go?
Slideshow will go on this guy.
Okay. I will need to borrow you your.
My stick? Okay.
I lost mine.
Do you have the plug-in?
And apparently my laser pointer. Oh yeah. It's over here.
Oh, okay. Mine doesn't accept those anymore unless I have the adapter.
Rachel, if we're recording Blue Jeans, we don't need to record it on a separate camera too, do we?
I don't think so. We've had both. We've usually just had this set.
Yeah. Just from and then Blue Jeans is recorded.
Yeah, I think maybe we have had both cameras.
I don't see a need really.
No, everything will be recorded off this presentation plus audio and video.
Yeah.
So there's no point using camera. It's just redundancy, really.
It's more editing for you guys.
Yeah, exactly.
And so the camera's really just to capture the presenter, right? Because the slides are shown regardless.
Yeah, so you see the slides and then just a little picture of the camera.
Right.
Once we actually start sharing.
Right. Are you gonna walk around, Luke, or do you kind of?
Yeah, probably a little bit.
A little bit?
Not a huge amount.
Well, the way the camera's facing right now, it's perfect.
Camera,
Yeah, that'll work.
See that camera?
I can stay within that.
Just so she's not.
Okay, so you've loaded? You can show black.
Oh, it's called night mode.
Oh, the
It's
night
the
mode.
new OS.
Yeah. I can load up.
Here I am right there. 2019-0108.
Okay.
Oh yeah.
So Chelsea will introduce you to the series.
Perfect.
Just like 3 of those slides.
Yep.
And then we'll hand it over to you. And by no means did you take up the whole time, but you have it.
I could. I don't want to.
We tend to use an hour for it.
Yeah.
Which one am I looking for
You
here?
want, okay.
So mine starts with 2019.
Luke,
Right there.
and then mine's buried. I'll pull it out.
Okay. I already checked it for you, so you don't have to worry about forgetting it.
I've got some water here, and I probably will give myself some coffee.
Okay, which one's first?
Mine, fall.
There.
Both sharing both presentations.
I'll just share PowerPoint in general. Then basically
Yep.
switch between both, you'll be fine. So you want this other guy?
Yeah.
We'll go start. I'm gonna put the video here at the top and just
Minimize it?
Yep.
Yeah.
And I guess the other thing is stop recording too.
Oh, how do we start this?
Okay, so you guys are done.
Because I've never done that.
Yep. If you guys are done, you just click up here to stop sharing.
Yeah.
And it'll basically bring you back to Blue Jeans. You got an option to stop recording here in the corner.
Oh, okay.
Okay. So when you guys are here into share screen, I guess it automatically.
It's got a green outline we're sharing, right?
So I can't.
It's a respiratory muscle training device. That's to improve endurance of breathing muscles.
So if you guys decide to move this video around,
We're testing.
right, let's say you want action this side,
It's kind of why I'm
kind of thing,
showing.
right, and hide it. Basically, it counts as a separate window, so there's like no control of your PowerPoint right now.
Talk to someone.
Okay, so you have to come
Oh.
back and click back onto the PowerPoint.
Divers.
Have
Gotcha.
control.
Okay.
That's the same thing. If you say, oh, I don't want it there anymore, I'm gonna move it here. You're like, hey, once again, you lost it, right?
Totally figured
So
out.
look at it as two separate windows regardless of what's on the same screen. Okay, so you just have to click back onto this.
Good, how you doing, mate?
Same thing when you're just like everything out of presentation, when you're ready to swap,
That was your quick
just
trip?
press the escape, go back to the normal PowerPoint video window,
I was ready to come back.
Free.
and then you just switch
Have
between.
to get home.
A little chaotic.
Can you be sure how to do that?
Yes, well, you know how to move PowerPoint to PowerPoint, right? I'm teaching.
Yeah, maybe, yeah.
It's all being shared right now.
Okay. Yeah. So basically, just escape out of there, go back to PowerPoint. So this is escape, and bring it back here to this view, and you just go down here, start your next one, and then just hit 5 again to start.
Like wedding planning,
Right.
like just
So if you see this green box around the window, that indicates we are sharing PowerPoint. This guy here, you can hide, he's actually part of PowerPoint, but you'll still have control of your slides. It's this black guy over here.
The other window. Okay.
So if you guys want to see anybody who's connected, just press this arrow here beside it, and it drops down the video.
Okay.
Okay, this is saying no one is sharing video. That's basically the people connected online. There's no one, any video online except for us. As soon as someone else connects, their video will technically show up there.
Oh, yes, right.
Right, so let's press
Yeah,
that.
unless they turn it off.
Click back onto the PowerPoint.
All right.
Guys, ready to go. Obviously, this is where your sound is going to be picked up for the people online. The further you guys get away from it, the less sound you guys are going to be projecting to
Okay.
people.
Yeah.
Especially if you turn your back to it, the top of this part of the room, you're not even talking to the box anymore.
Okay.
So you keep that in mind that this is where the people are going to be hearing you online.
Can we put the box there at that end?
Yeah.
That'd probably be better.
Bree's gonna monitor the blue jeans if there's any issues.
I can get on
Cool.
to it.
If she can get.
That's the only thing I would tell you guys concerned about, is the sound.
Okay, great. Do you want to get coffee with you?
Yeah, sure.
Just 3 slides.
Yeah,
Some
that's fine.
sponsors and then a schedule for the semester.
And then introduce group and then those highlights, yeah.
You guys said are done at 2:30ish?
We'll be probably done before then.
Okay, we'll come back between 2 and 2:30 then.
Yeah, probably around 2.
Luke doesn't not mess around.
Waiting for 2 to know, no, come through.
So, how? It shows the presentation, but not the video.
I will introduce you briefly.
Yeah. Great.
Is there anything you want to say?
Video camera, because it's this way,
Yeah.
when you're looking this way, always look at the back of your head.
Okay, I will attempt to,
Or just
Kind of like
stare at the
this,
camera
or
the whole
this.
time?
If you look at this, like, really,
Sure.
maybe, you know what, maybe just move this.
So you're monitoring regions, right? So if you see anybody pop in with a message like, I can't hear.
Yeah.
Okay. Mm-hmm.
Okay.
All ready?
Yeah.
Hi, everybody. Happy New Year. Thank you all for coming to our first Health Research Institute seminar series of 2019. So we want to thank our sponsors who provide some funding for this ongoing seminar series, as well as highlight some of the great upcoming presentations this semester. In February, we have Sarah Gray, who's another very proud member of our organizing committee for this series. In March, Dawn Hemingway, who's from the School of Social Work. And then in April, we're going to showcase some of the great work done by our undergraduate honor students in the School of Health Sciences as well. But today we have Dr. Luke Harris, who is an associate professor in the School of Health Sciences at UMBC. here. And he is going to talk to us, I don't have the title off the top of my head, but he's going to talk to us about the great work himself and his fantastic students do in his near-infrared spectroscopy lab. And I'm going to switch to slides.
Okay, thank you very much, Chelsea.
You're welcome.
Okay, so thanks to those of you who are here in person and also those who are connected by phone or Blue Jeans. If you have any problems hearing or seeing, or you're having trouble with the electronic thing, we do have a person in the room who's monitoring the Blue Jeans connection. So feel free, I think, to just pop is there like a text message function in there? There's an option to send a message textually that we can use to sort that out. The other thing is I'm going to do a couple demonstrations. One of them will actually require me to switch the screen to a different computer. I think what that probably will mean is that the people connected via Blue Jeans will lose their video feed temporarily. So we're going to try to keep that to a bare minimum. But for those in the room, it'll give you something a little bit extra to see. I'm just presenting from a computer that I wasn't able to install a separate piece of software on that I need for the demo. So without any further ado, so I had kind of an awkward title because I had promised that I would send a title and a summary of my talk by a particular date, and I forgot until the day after that date. So I kind of threw this together. So obviously the thinking part refers to brain, and the meat part refers to muscle, and the bones part refers to bone. But there's also kind of a message in here that's buried a little deeper. One of the things that I reflect on a lot when I'm teaching, for example, is that I'm a generalist. I have a lot of different interests, and those arose from a variety of personal experiences. And so I guess as one example, I have long had joint problems and some arthritis since before the age of 20. And so I'm really interested by the skeleton and what the skeleton does. I went to do a PhD in neuroscience, so I thought I was going to be studying the brain and spinal cord, but as things transpired, the PhD project that I planned on originally didn't really work out, and I ended up working on muscle, which was totally an accident. It's not what I wanted to do, but I love muscle now. So one of the things that happened through all these years was that I started to see connections among these different systems, and it may seem kind of strange to say it, but there are connections that we can draw between the function of the brain, the health of the brain, and the function and health of bones to some degree. And I'm not going to span that entire gap today, but I realized at kind of a critical moment, I think, during my graduate studies a couple of years before I came to work at UNBC, that there were ways that we could integrate across these various systems and look at the well-being of people as kind of an integral whole, which sounds very lofty, but it's kind of what I shoot for when I do research. So we have a picture here, and there are a couple of people in the room who will remember this slide from a presentation I've given somewhere else, but here is an instrument. What is the instrument? I hear some whispered responses. The question is non-rhetorical, so I am looking for an answer. Okay, somebody says CT or MRI. Anybody want to try to refine that answer a little bit? It's an MRI machine, that's correct. So what can it do? Take pretty pictures of tissue. What kind of tissue? Soft tissue. What kinds of soft tissue? The brain. So, I mean, MRI is probably best known for its ability to image the brain, but it can do a lot of other things. So I've been in an MRI machine a couple times. Once was to look at my knee, once was to look at my ankle. So in those particular cases, they actually were looking for issues that were associated with not just soft tissues, but actually hard tissue. The extent to which it can do that depends on how you use the instrument. So I got this from Wikimedia. Here's another one. What is this one? Somebody whispers a CT scan. So yes, this is a CT scan. What does it do? Muscle? Anything else? Bone? It can image basically all the same things that an MRI can, but it does it in a different way. And the utility of them is dependent upon the properties of the machine and the type of, or the configuration of how it physically monitors what's going on with the body at a particular time. So this is a generalized floor plan for a room in which X-rays are conducted. And one of the questions that we might ask ourselves looking at it is, what do we notice about the way in which this room is laid out? So we have the actual X-ray emitting and receiving device, or the X-ray emitting device here. There are some cubicles along the side. Difficult to know exactly what those are intended for. We have an additional patient bed over here, which presumably is for preparing patients in advance of actually having the X-ray done. And then we have an area over here with a sink, which we could assume is a place where we would wash hands. And then we have the control desk. So in terms of that overall layout and the relationship among the different components of this space, what do you notice? Exactly true. The operator is outside where the X-rays are. So we don't see that here, but a CT scanner actually uses X-rays. So that would be the same situation. And then if we go back further to the MRI, one of the key things is that we have both the patient and the operator in the room at the same time. Now, normally in an MRI situation, you wouldn't have an operator or a technician in the room that the patient is in. They'd be watching a computer screen. But this implies to us that there are certain risks associated with performing this particular type of imaging. So what does all of these have in common? What do all of these have in common? Imaging, what else? They're non-invasive. You go into it. Well, to a certain extent. I mean, with the X-ray, it kind of passes over you. You get, of the person, right? Right. You get to see the inside of the person. Anything else? Shorten your lifespan. They shorten your lifespan. Is that true for all of them? I mean, I think we'll probably pretty safely say that MRIs don't shorten your lifespan, but I also suspect that nobody's tried to collect longitudinal data on that. We don't really know. It hasn't been around long enough. X-rays certainly will. Sufficiently so that when I went for a CT scan recently, I asked the tech, what is actually the radiation dose? What are the potential effects of this on me? And the tech kind of said, oh, well, don't worry about that. But I've had conversations in the very recent past with GPs who say things like, well, when we have a patient who's in a particular type of condition, even if we have a very strong sense that there is diagnostic utility of using a certain type of test or imaging, we may not if the benefit is insufficient to merit the risk in that situation. So,
what do you mean by shorten your lifespan?
Exposure to radiation, in theory, could shorten lifespan. I mean, I think Tammy said that somewhat jokingly.
You know, well, a little bit, because there are some scans, for example, you have to drink dye, and for some people, it can actually cause really bad reaction, right, from drinking that.
Yeah.
So it has risks and it could shorten. Yes, but right. It doesn't shorten your lifespan, but
correct. It doesn't shorten your lifespan by definition, but some of them may carry risks that you have to think about. Actually, the first time I ever went in an MRI machine, I was a graduate student. I decided to participate as a volunteer for a study, and at that time I had pierced ears, and I had to take my earrings out. Don't want metal in your body when you go in one of these machines. So anyway, all of these instruments have certain things in common, and one of the big ones that we haven't mentioned is that they actually each require a room. They're big. They take up a lot of space. So they're large, they're non-portable, for the most part. Now, there is a so-called portable CT scanner in operation on Haida Gwaii at the moment, or at least there has been in recent memory. It's in a flatbed. It's in a semi-trailer, and they drive it around the island. And this was a pilot project that was initiated by one of the doctors there in order to try to determine whether or not they could do improved cancer screening before making the decision of whether to send somebody off island. Because there were a lot of people who simply weren't going off island because they didn't want to leave home. So that was addressing a barrier. But you know, if you think about the cost and the logistical challenges of putting one of these in a semi-trailer and sending that around a geographic area, there obviously are disadvantages. They are potentially hazardous. And then, as I said here, they are not necessarily great at showing changes in human physiology as they happen. You get a slice, you get a snapshot in time. So then the question is, what is NIRS? And NIRS stands for, I'm going to give you part of the answer, NIRS stands for near infrared spectroscopy, which is the type of instrumentation that we primarily focus on in my lab. And I know that there are some people in the room who definitely already know what near infrared spectroscopy is because they've heard me talk about it ad nauseum. Oh, you're not one of my students. Anybody want to try to answer that question just based on the name near infrared spectroscopy? There are no takers. Okay, how about oh, Sarah.
I'm not sure what your question is. Like it in real time?
Any kind of answer, actually. Just what is NIRS? Does anybody know anything about it that they're willing to hazard a guess at? So you have said changes over time in a patient. What do you mean by over time?
Like
Right, blood oxygenation over time. So that is actually a very good summary of the kind of thing that we're interested in. Oxygenation is sort of one of the two main variables that we're interested in when we use near infrared spectroscopy. So it's a tough question if you don't look at and think about this stuff every day. This is a NIRS instrument on somebody's forearm. Now, I think this is the point at which I will do a very quick live demonstration for people who are in the room. Now, do you know, Rachel, if I switch screens, will the people on Blue Jeans lose the video feed?
I don't.
Okay, well, we're gonna find out. I'm actually gonna switch to a different computer very briefly, and I'm gonna ask Kavala to come up and help me very quickly with something here. Do we have video feed, Bree?
We have video, and then we still just see the other. We don't see that.
Okay, all right. So this will be very brief, but hopefully this will be large enough that those people who are joining us via video conference can actually see. Trying to find my mouse here so that I can actually resize this window. Here we go. That's good. Okay. So Kavala, who's standing up here with me, is a fourth-year undergraduate student in the health sciences biomedical degree, and she is doing her honors thesis in my lab under the supervision of Tammy Clausen-Ross, who's also in the room, with co-supervision from me. And basically all she's going to do is she's going to put a NIRS instrument on my forearm, and I'm going to demonstrate a very, very simple principle of what it is that we can measure. If you can just put it over with the table going this way. Yeah, perfect. So I've had this thing charging up for a while, and I'm going to do something either very brave or very stupid for the purposes of the demonstration, which I'm going to actually unplug the cable because I'm connected wirelessly to the computer that's running this instrument at the moment. And actually, Kavala, if you could be the person to hit start and stop, that would be awesome.
Sure.
Since I can't really see what I'm doing. So.
Where are the start and stop?
Oh, it's up here. You're going to have to crane your neck over your shoulder.
on the laptop. No. There we go.
Yeah.
And this
Start is the first big button.
is right there.
Perfect.
Okay, do you want me to start now?
Yep. So what we have here are 3 lines in 2 different channels. And each of these channels shows a depth of penetration of our signal into the tissue that is different. So channel 1, which is on the top, is a more shallow depth of penetration, and channel 2 is a deeper depth of penetration. So basically, light is being sent by the instrument into my muscle at the moment. And what we're seeing is a red line that shows us how much of the blood that is flowing through that muscle is oxygenated. The blue line shows us the component that is deoxygenated, and the green line shows us the total. Take a breath. Well, you know I could. So usually we don't see that kind of distant effect, usually, but what I'm actually going to do, so you can see that the red is coming down actually and the blue is going up. And one of the reasons for that is that actually just from holding my arm up, I'm getting a little bit of, so the oxygen is being consumed. However, what we can see that's a much more dramatic effect is if I actually contract that muscle. And then you can see the blue is going up quite dramatically. The red is going down quite dramatically. So I'm really deoxygenating the muscle tissue right now. And the green is actually fairly stable, going down just a little bit. And if I release, then we get an immediate rebound and a return of the green and the red toward kind of the baseline, and the blue starts to come down again, and Cavala can be paused. Okay. So I don't know how well the people who are visiting by a video conference can see this, but we get this increase in the blue, which is the deoxygenated part. We get a decrease in the red, which is the oxygenated part. And then the green is kind of fairly stable. The reason why we care a lot about the green is because, thank you, Cavala. The green actually tells us the total volume of blood in the muscle. And under those specific, if we want to call it an experiment, under those specific experimental conditions, the muscle was not actually squeezing hard enough to push the blood out of it very much. I'm going to switch back to the main PC now. So the inspiration for that is this study from Babak Shadgan, who has been a friend and colleague of mine for about a decade now. We actually first met in 2008, so it's actually been more than 10 years, which is kind of hard to believe. But he did this very elegant experiment with Olympic wrestlers at the 2008 Beijing Olympics. And we see here essentially the same thing that we just observed. So this is over a much longer time period, but the wrestlers are doing first 10, then 30, then 50% of what we call the maximal voluntary contraction, or the MVC. So this is the most amount of force you can generate with a particular muscle or muscle group, and then we work at a percentage of that. So they've previously calculated this MVC based on how much grip strength the wrestler could produce using this grip dynamometer down here, and then they're measuring NIRS parameters at the same time. So one thing that we see in this chart that we didn't see in the real-time chart just now is we see something called the TSI, or tissue saturation index. This is kind of like humidity. If you've ever thought about relative humidity, we see that relative humidity can swing between, let's say, 5% and 100%. If we have relative humidity at 100%, that means that the air in the local area where we are is holding as much humidity as it possibly can. Probably just rain. So in the tissue saturation index, the closer we get up towards 100 on this y-axis, the closer we are to the maximal potential amount of oxygen that tissue can hold. So in cases like this, where we see drops in this so-called TSI, or tissue saturation index, what this is telling us is that the muscle is losing oxygen relative to its potential 100, or potential max. So we see here concentration units which tell us how much we have of this, in the case of red, oxygenated hemoglobin, so the protein in blood that carries oxygen, or the deoxygenated hemoglobin, when that protein is not actually bound to oxygen, and then the total, which is actually just the arithmetic sum of the oxygenated and deoxygenated. Now, it still kind of blows my mind sometimes when I think about this, even though I've been working with NIRS for about 10 years, that something as simple as this arithmetic sum, this green line, can tell us something so valuable. It has been repeatedly validated as a good and reliable estimate of the volume of blood in the tissue at a given moment of time. And then if we look at changes over time, we can interpret that as flow. So if we look at a region like this, where it's steadily decreasing a little bit, we can interpret that fairly reliably as a steady, small decline in the blood flow through the tissue. So what does this mean when we look at this experiment and we think about what happens when we contract muscle? Well, the 10% MVC leads to a decrease in the green trace, which is our blood volume, so the amount of blood in the muscle. And the red tells us that there's a decrease in the oxygenated part of that blood. And the blue tells us the opposite, which is that there's an increase in the deoxygenated part, which seems fairly straightforward. Those differences get larger when we go to 30% of MVC, and they get even larger when we go to 50%. Now, the part that's really interesting and important from my perspective is that then we go over here and we look at ischemia. So we have here a sphygmomanometer, or a blood pressure cuff, wrapped around the upper extremity. And so at a particular point in the experiment, they inflate that blood pressure cuff to the same pressure that you would use during certain surgeries to eliminate blood flow into and out of the liver, so that we can prevent blood loss during invasive surgery. And the reason we care about this is because we actually see an opposite response. So if we use an external pressure, an external manipulation to compromise the flow of blood into and out of the tissue, we get a totally different response from when we voluntarily use that tissue. And what's quite profound here is this green trace. Because the green trace shows us that even as the muscle is working, it is eliminating its blood supply. It's pushing the blood out of the tissue. Now, the thing that's counterintuitive about this is that the tissue needs oxygen in order to do its work, or glucose. It needs to eliminate waste. And so when Babak and I were talking about this experiment after he came back from China, we had this conversation where I, knowing nothing about NIRS, I'd never worked with it before. This was basically my introduction to it when he showed me his pilot data. I said, you know who would probably have better ischemia tolerance than wrestlers? Cyclists. And he said, no way. Cyclists don't have forearms that are as strong as wrestlers. And I said, but it's not about the strength of the muscle. It's about how resistant it is to prolonged ischemia, prolonged lack of blood flow into the tissue. Because cyclists who are competitive, they're generating grip with their forearms, leaning hard on the handlebars for extended periods of time. If they're real endurance cyclists, maybe 3-4 hours. So we did this on some cyclists, and actually, we saw less effect on the oxygenation of these tissues and even less effect on the blood volume of these tissues in the forearm muscles of the cyclist. So it tells us something really interesting about how muscles can adapt through training to the kinds of pressures that we put on them. So this is kind of my way of through demonstration saying, this is what NIRS is and what it can do. It can tell us about the blood supply to a tissue, and related to that blood supply, what the metabolic status is. And if we look at specifically this tissue saturation index, which tells us how well oxygenated is this tissue relative to its potential maximum level, what we see is that completely compressing the vasculature as much as is safe to do in an experimental setting with an external cuff, we don't even get the level of deoxygenation that we get from 10% of the maximal potential force that muscle can do. So an active exercising muscle is able to very easily eliminate its own metabolic supply as it works. And that tells us something very important about the consequences of exercise and how muscles may adapt to exercise over time. Now, I'm probably going to have to skip a bunch of slides here because in tip...
Can I ask a question about that?
Yeah.
But you haven't told us how they function when they do that. You said it's really interesting. When they get rid of weight, but they're So what's happening?
Okay. So there's kind of two answers to your question. If this period of sort of voluntarily exercise-induced ischemia were to be very prolonged, it would adapt. So one of the things that would happen is that the muscle would start to fatigue a little bit, and it would actually begin to release some of the pressure on the blood vessels so that more flow could come in. So that's one answer, is that over time, as the exercise progresses, the endocrine system will release hormones, the autonomic nervous system will kick in, and you'll actually induce vasodilation to that tissue so that you can get better blood flow. The other answer to your question is that we have a bunch of different supplies of energy. So in the case of muscle, you have intracellular glycogen stores where you have lots of sugar. So you kick in glycolytic metabolism and you start to produce ATP from sugar. We also have intramuscular fats. So that's not stored within the muscle fiber itself, but it's nearby. And the muscle is able to locally access those adipose reserves. So over very long exercise duration, muscles are able to do that. But the other part of the answer to the question is that usually when we're doing exercise, with the exception of examples like somebody leaning on bicycle handlebars very hard for a long time, we don't have static contractions that endure for, let's say, hours, usually. We have contraction, release, contraction, release. There's actually a third part of the answer to your question, though, which doesn't have to do with metabolic supply. What it does have to do with is the relationship of exercise to aging, which is a tangent I didn't plan to go on, but it's actually very interesting and useful to think about. So we undergo oxidative stress as we age. And this oxidative stress has a number of sources. Maybe we eat too much pastrami. I know I do. But there are lots of ways in which we can induce biochemical stress in our tissue. And by undergoing bouts of ischemia voluntarily through exercise, we can actually condition our tissues over time to be able to withstand that kind of metabolic stress. And so one of the effects of doing exercise a lot over extended periods of time is that our muscles upregulate the enzymes and biomolecules that we use to combat oxidative stress. And so there were data that were published very recently. I think I heard about this just before the Christmas holidays, but there were data published very recently that found that people in their 80s who had maintained, let's say, an exercise regimen of an hour, 3 1-hour sessions per week over many, many years had muscles that were biochemically, functionally, structurally indistinguishable of those of people in their 20s. So the long-term benefits of exercise, which is not the point of this talk at all, but the long-term benefits of exercise, which to some extent we can study using this type of equipment, are profound. Okay, I'm going to move ahead a little bit here because I'm super slow and we need to get to Kavala, who's going to talk to us for 10 minutes here in a moment. Okay. You guys know how I can skip through many slides at once here? Oh, no? Okay, well, you're just going to see me skip through an excess of slides here. So the basic principle of NIRS is that biological tissues are transparent to light. And near-infrared light, which is what these instruments use, is non-visible, but it has similar properties. And unlike ultraviolet light, which is at the other end of the electromagnetic spectrum in terms of light, just beyond purple, that's ionizing radiation. We know if we spend too much time in the sun, the ultraviolet light can lead to skin cancer. Near-infrared light does not do this. So we can use it relatively safely. Skipping through an excess of animations here. This is a schematic. Again, this was drawn by Babak, my colleague. So we have an emitter which sends out this near-infrared light into the tissue. These arrows are meant to indicate that basically this light can go any direction within the tissue. Right, so it's not very common that this happens, but every once in a while I'm talking to somebody and they say, oh, well, how does the light know to go from here to here where it's detected? How does it do that? Which is kind of like saying, if I take an ibuprofen, how does the ibuprofen know to go to the headache? Well, it doesn't. It goes everywhere. And the molecules of that drug that end up in your brain, or the vasculature associated with the brain, they have the effect. So the photons of light go everywhere in the tissue, and the ones that happen to get scattered along this approximate path from emitter to detector are the ones that we measure. And basically, without going into any more detail than this, we can compute the differences, sort of the differences in the properties of the light that we detect here compared to the light that we know we emitted here, and we are able to determine the properties of that light and say how much of the blood is oxygenated versus not. Okay, there are some pictures of hemoglobin and myoglobin, which are the proteins that bind oxygen, which we measure. Another schematic that was drawn by my PhD student, Tim, that I obviously don't have time to talk about. A question I get often, which I frequently overlook, is how is it that we can use NIRS to measure differences between something like hemoglobin bound to oxygen and hemoglobin that's not bound to oxygen, because they should be very similar. Actually, this shows us wavelengths of light. And if we get down here, we're actually talking about visible light. Up here, we're still talking about near infrared light, so the light that is just beyond the light we can see. And here we have the specific extinction coefficient, which basically is a fancy way of saying how much of the energy of a light at that wavelength is absorbed by the tissue. And so this shows us that absorption value for the oxygenated hemoglobin, HbO2, and this shows us the absorption value for Hb. And the reason why they put Hb and HbO2 in these particular diagrams is because when we use NIRS, like the instruments that I used for the demonstration just now with Cavala, what we actually are measuring is light emitted at two different wavelengths. And those wavelengths are selected such that the difference in how these two molecules absorb those wavelengths is relatively great at those wavelengths. So here, oxygenated hemoglobin absorbs light of this wavelength, near 1,000 nanometers, much more powerfully than deoxygenated hemoglobin does, and vice versa. And so by comparing those two differences at those two wavelengths, we're able to determine how much blood is oxygenated and how much blood is not. Now, in principle, this is a very simple method to use. And before I forget to say it, one of the things I wanted to say as part of this demonstration is that we have this tiny little thing, we can put it on a piece of muscle tissue, we can measure a change in real time while it's happening, let's say in a patient who's doing, I don't know, a walking intervention on a track. You cannot do that with MRI. You cannot do it with X-ray. You cannot do it with CT scan. Are there disadvantages? Absolutely. But one of the reasons I got excited about this instrumentation originally, and one of the reasons why I'm still excited to use it as much as possible, is because we can take it where we want. And Chelsea, who introduced me earlier, supervises a student that I co-supervise, Mike Eden, who took these instruments with admittedly results that we are not sure of the outcome yet. We don't know how useful they really were, but the point is that he took them into wildfire fighting camps and measured directly from wildfire fighters over a couple of summers. And for us, for me anyway, that was a really big deal, because one of the things I wanted to do was say, we can actually take these instruments out, measure from people who may be undergoing some kind of health challenge in the location where they are experiencing that health challenge, rather than having somebody who let's say experiences health challenges and then they have to come into a clinic somewhere, and now they're removed geographically and temporarily from that health challenge. So that, to me, that's the potential of instruments like this. Okay, lots of other scientific stuff that we don't need to talk about right now, et cetera, et cetera. Some goals of what we do in the lab. I guess it's probably worth mentioning what these are. We are really interested in how we can improve brain function, respiratory function, and also the overall well-being of both the musculoskeletal system and the neuromuscular system throughout, and I've used this word very intentionally, the health span. So we're not just talking about people living for a long time, we're talking about people getting to sort of a near end of life stage and still being healthy enough to enjoy that. That's really important. So currently we're really interested in young and healthy individuals in particular, and sort of what are the fundamental functions of the brain, the neuromuscular system, and the musculoskeletal system, as well as the respiratory system. And some of the potential applications of what we do are for people with acquired brain injuries like stroke, also things like chronic obstructive pulmonary disease, and I have a particular interest, long-standing since my undergraduate degree when I first started doing serious research, spinal cord injury. Okay, I'm going to skip through a few slides here that relate to something that's very interesting but not easy to explain given the time pressure I have put myself under here. However, I'm going to, well, you know what, I'm going to come back to this. Why don't we get Cavala to come up, and she is going to talk to us a little bit about a project that she has been developing for a number of months that she's very excited about. And then I will decide after that what to continue talking about.
I can keep mine short, so I can cover lots of other stuff.
I'm sorry about this slide, Cavala. I don't know what's going on with this.
Oh, that's okay. It's got all the information on it.
Cavala had a very beautifully set up presentation with lots of incredible colors, and for some reason we have now lost it. So apologies to Cavala.
Survive. Okay. So as Luke said, I'm Cabela, and today I'll be talking about my project that I'm doing for honors. I've been working on it for almost a year now, not necessarily this project, but I also did a URE project that was related to this. So this project is entitled An Investigation of Acute Moderate Exercise's Effects on Cognition and its Neural Correlates. So that may sound confusing, but the basic premise of my research is to try and see what happens to cognition after exercise is complete. So first, some background on just the general knowledge surrounding this topic. Research has found that exercise increases cognitive ability in both the healthy population and in other populations, such as people who have dementia. They've studied this during exercise. Another thing they found is that increased cerebral oxygenation has been shown to increase cognitive ability during exercise. Also, stress hormones released during exercise can also impact cognitive ability. So what do all these things have in common? They are all during exercise. To my knowledge, I've only found 3 studies published within 2017 to 2018 who specifically looked at post-exercise. So this is a very new area of study. So my research will hopefully look at what happens after exercise. So overall, there's a couple different steps to my project. So the bottom line is that I want to see what happens to cognition after exercise. And I'm also going to be looking at several different physiological factors to see how those change during exercise and how those could also potentially affect any changes I see in cognition. So the factors that I'll be looking at are cerebral oxygenation and the stress response, which includes cortisol and norepinephrine. Whoa. Okay. So,
all I'm getting is poke.
Yeah, there's also post-exercise. Okay, so in the first square over there, was supposed to be, I'm going to do my baseline measurements. This is the procedure, by the way. I'll be doing my baseline measurements, which will consist of cerebral oxygenation measure, stress response measure, and also different cognitive tests. So once I've done my baseline measurements, my participants will do 20 minutes of mild aerobic exercise, which is the green box up there. And how I'm going to do that is they're going to do 60% of their heart rate reserve. And heart rate reserve is a calculation of resting heart rate and maximal heart rate to see what potentially is their maximum exercise capacity they can do. And then we calculate 60% of that, and they do that for 20 minutes. So then these next 5 boxes are the post-exercise tests. So what I'm going to do is immediately after exercise, I'll do another cerebral oxygenation measure, stress response measure, and 3 different cognitive tests. The cerebral oxygenation will be recording the whole time, but at each point where I collect the other data, I'll put a mark in there so I can see exactly where that is. So in each measure, I will be doing 3 cognitive tests, the stress response test, which I'm doing through saliva samples, and cerebral oxygenation. So now I'm going to talk more about those measures because each one has several different steps to it and why I'm using them. So first, cognitive function. I decided to specifically use executive function measures. Executive function is basically your higher order processes. So you need your executive function to plan. There's 3 different components of executive function: inhibition, working memory, and mental flexibility. I'll be using 3 different cognitive tasks to target each one individually. Also, executive functions utilize the prefrontal cortex, which is why that's what I'm going to be measuring cerebral oxygenation from. So, how am I measuring cerebral oxygenation? As Luke talked about, I'll be using, don't know what happened to that title, near-infrared spectroscopy, which I'll go over very briefly. But basically, it's non-invasive and easy to use. It's here at UNBC, so students have access to it if they wanted to get into research. Basically, it uses near-infrared light to measure cerebral oxygenation in the brain. Specifically, I'll be using the prefrontal cortex because that's where my executive function tests are based. Next, we have the stress response, which is one of my physiological measures that I'll be using. So exercise is a physical stress that activates the stress response. When the stress response is activated, it releases cortisol and norepinephrine. Both of these hormones can be measured in saliva in real time and fairly accurately, which works out quite well for my project. It's also been linked to cognitive performance. There isn't a specific level of hormone that's correlated to either high or low cognitive function, as I can tell. So that will be another area of study if I could correlate my microliters of cortisol and norepinephrine to my cognitive function. Okay, so now that we know what I'm doing and my individual steps, why is this research important? So I have two different sections to this, academically and then in the real world application. So academically, this is fairly new research, both in the aspects of studying post-exercise in cognition and with NIRS. So the three studies I found used only one test to look at cognitive function after exercise. My study will use three different tests that assess all the aspects of executive function. So that will be a new area of research and hopefully add breadth to the literature. I also haven't found studies that focus on cerebral oxygenation after exercise, so that will also be a new area to add to the literature. Also, since I'm doing cognition and different physiological correlates, I'll hopefully be able to link the cognition scores to either cerebral oxygenation or stress response or both, because any fluctuations in those could correspond to fluctuations in cognitive performance. So what does this mean for the real world? So for people like me, I'm a university student. If my study does in fact find that those effects on cognitive function during exercise are also applicable to post-exercise, I could go for a run before I take a test or study after I exercise. Also, my eventual goal is to work with a population who have dementia to improve quality of life. And they found that executive function declines during dementia, which is one of the reasons why I picked executive function in this study. Also, they found that exercise improves people's cognition who have dementia, but people don't know what happens after exercise, which is hopefully where my study will fill in. And it does mean that my study shows that increased exercise increases cognitive performance after you exercise. Then people could schedule mentally draining tasks after exercise and hopefully improve family visits and quality of life that way. So I hope that this research goes well and it can be applicable to both real life and in the literature. Thank you. I'll pass it back off to Luke.
Do you like to have questions about?
Sure, we can save it to the end too, it doesn't matter.
No, go now.
Okay.
So with dementia, we know that it progresses first.
Yes.
So let's say that exercise does show to improve executive functioning.
Mm-hmm.
What is your thoughts on it saving off the actual progressive nature of dementia?
Yeah. So there is a huge body of literature around this, and some studies have found that there's mild cognitive impairment, and then that has been shown to lead into dementia. They're kind of wishy-washy whether it's a precursor or the early stage of the disease. But they found that people who have mild cognitive MCI, there we go, have lower levels of white matter or gray matter. Can't remember which one, but one of those. And then if they get them to exercise, that brain matter increases, and so does their cognitive performance. So in that study, it looks like exercising could prevent dementia, but I'm sure that there are so many other factors that need to be looked at to make it a definitive prevention strategy. Does that answer your question? Okay. Any other questions? No? Okay. Thank you.
Thank you, Cavella, very much for coming in and talking about your work. Okay, so I've talked about muscle a little bit, although not so much the work we do in my lab, but since I promised you brain, muscle, and bone, I will, doesn't seem to be an easy way for me to, oh no, I did not want to do that. Oh, yeah, beauty, okay. So I'm gonna skip. These are really cool data that I don't have time to talk about. Tim's awesome work, and I left that title there for Tim's work because I thought it was hilarious. That's not the sort of thing he would ever say. He clearly intended for me to insert something different there. Whoa, and it's in there twice, apparently. Okay, boom. So this is entirely new, and one of the reasons I really like to talk about this is, well, it's twofold. So the grad student who's working on this couldn't be here today, but he has taken on an enormous and frankly quite daunting challenge to try to measure something in bone that has never been measured before, and that I was very open with him from the get-go. We might not be able to measure. We really don't know if we can do this. But I think we can. So I said to Colton has an undergraduate degree in physics, and I said, Colton, I really want you to try to undertake a fundamental study of the optical properties of bone using near-infrared light, and then apply that to trying to look at some functional outcomes. So he did an extremely preliminary pilot study as part of a course that he did last year. And he was really interested in knowing whether we could use one of these portable instruments in a practical setting to measure side-by-side metabolic properties in bone and muscle in the same leg during both dynamic and static exercise. So we use NIRS to measure both from the shin bone or the tibia and also from one of the calf muscles, the medial gastrocnemius muscle. And participants did static and dynamic calf raises. So this is a really simple and kind of, honestly, a little bit crude schematic, but this shows us exactly what we would expect to see. So here we have a decrease in oxygenated hemoglobin in the muscle over the course of a static calf raise exercise. So participants were in a calf press machine, they stood up, and they held the contraction at 50% of their maximal voluntary contraction, either for a minute or until they were exhausted and couldn't hold it anymore. So a minute at 50% of your MVC on a calf press machine is a lot. It's very difficult. And so this is exactly what we expected. There's a lot of decrease in oxygenation here. Then we see a relatively stable blood volume, but it goes down a little bit. And then we see an increase in the deoxygenated hemoglobin. So this is exactly what you would expect from demanding exercise in a muscle group. Now, what's interesting here is we measured a different parameter from what I've showed you so far in this talk. This is called HB-diff. And this is a unique parameter that we've started measuring in muscle that tells us how much oxygen the muscle is actually consuming. So we're not just measuring a decrease in the amount of oxygenated hemoglobin, we're actually now directly measuring the oxygen consumption by the tissue, which is something relatively new in our group. So obviously we can see that this muscle is actively using the oxygen that is disappearing from the tissue. What was really interesting is that when he looked at bone, he saw something entirely different. This is the deoxygenated hemoglobin, this low flat trace here. And this, I don't know which is which anymore, I can't remember, but this is your total hemoglobin or your blood volume and your oxygenated hemoglobin and your HB-diff. Now, one of the most important things to notice right away is that regardless of what else is going on, your HB-diff is going up, and contrary to what you would think, if HB-diff goes up, you're not consuming oxygen, it's the opposite. It's almost like the muscle is de-consuming oxygen. We could almost interpret this, obviously incorrectly, as the muscle producing oxygen, which is obviously not true. So this is a physiological conundrum. What it tells us is that bone is responding very differently to this strenuous exercise, which is exactly what we would expect. Now, one of the things that I find very interesting here is that under this high load, there's an initial increase up to a peak followed by a steady decrement in these values. And from the perspective of blood volume alone, what I'm thinking is going on here is that you have capillaries running through the bone that are fed by a primary nutrient artery. That primary nutrient artery enters into the bone, spreads out throughout the bone tissue, but those capillaries are contained within rigid chambers of bone mineral that cannot expand. So in situations where your blood flow into the bone needs to increase, you need to have room for those vessels to expand. Now, what we saw earlier is that when muscle is exercising very heavily, it's squeezing blood out of its blood vessel. So what I'm thinking is going on is that as the calf muscle is working very hard here, initially, the blood that it's squeezing out is being sent into the nearest reservoir, which is the blood vessels within the bone. And then that blood slowly is restored out into collateral vessels that are traveling elsewhere through the limb. So this may not have anything directly to do with bone physiology, but we don't know. This is an initial attempt. We saw something that kind of makes sense in the context of what we would assume to be the case, and we'll have to follow this up with more comprehensive study later on. I'm going to go back very, very quickly and show you the thing that I'm the most excited about at the moment. I'm also very excited about Cavala's work. Don't worry, Cavala, but it's not finished yet. So we'll see what comes out of it. Okay, so this is from a student named Johnna, and I'm just going to show you very, very briefly the instrument that she worked with. This is called a spirotiger. And the idea behind the spirotiger is that it tells you what rhythm you're supposed to breathe at, and part of the air that you're breathing as you go on, that you're breathing in, is coming from the atmosphere around you, as we normally do, and part of it's coming from this bag. So you're breathing out into the bag, and then you're rebreathing from the bag. And there's a very cleverly designed, patented magnetic valve in here that adjusts to determine how much of the air you're breathing from the atmosphere versus how much is from the bag. And this is my best demonstration of how you're supposed to use this thing. I'm not good at it. So just over those few breaths, this machine didn't beep at me, so I was obviously doing okay. But the idea here is that we're supposed to specifically train the respiratory system to work more effectively and more efficiently. And what Johnna is doing that's very interesting and quite new is to look at the changes in blood flow through the prefrontal cortex, the same region that's in charge of executive function that Cavala is looking at, and also to look at the oxygen saturation. And the very brief take-home message here is that she found, very surprisingly, an interaction over 4 weeks of training that focuses specifically on the respiratory system. She found an interaction between the right hemisphere of the brain and the left hemisphere of the brain. And even if we don't know very much about brain function, we know we have right and left brain functions. These black bars show the results of an exercise test to exhaustion, where people rode on the bike until they could not go anymore. And that was before the training happened for 4 weeks. And these white bars show us what happened at the end of those 4 weeks. So what she sees is that over the course of training, for whatever reason, we see an increase in how much of the blood is flowing through the right side of the brain, and a decrease in how much of the blood is flowing through the left side of the brain. This is entirely new. It suggests that there's some kind of physiological adaptation in response to respiratory system training that is shunting blood from left to right during demanding exercise. This is a very statistically significant finding. The effect size is large as well. So we're fairly confident, even with, I think, only maybe 10 participants, that this is real. As to why it's happening, we don't know. I've speculated about it, but I'm going to hesitate to speculate at this point. But this is really, really interesting, and I'm really excited to pursue it further. And I'm excited for Johnna that she came up with something so cool and novel. So that's basically the end of my discussion about putting meat on bones. I hope that it was really interesting for you to kind of get that flavoring of the different approaches we're taking to looking at health and well-being according to kind of fundamental physiological function in different systems of the body. I have to skip through 39 slides. I don't know if I can do it. Here we go. I think I should. It would be best. Can we use it in a healthcare setting? Blah, 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 blah. Yeah, probably. Okay, so funding from all these various sources made the work that I presented possible. And thank you very much. Sarah. Yeah.
So the work that you showed on the bone, is that the first time that flow, is that right?
As far as I know, this is the only time anybody ever has measured blood flow in exercising bone. So yeah, it's the first time. Uh-huh.
But one of them is also
Right.
and I don't know if the mechanism for why you get growth with exercise, but actually blood flow to the bone itself.
Mm-hmm.
With very good that mechanism hormone as you blood. I just think it's quite interesting. Or because I'm not sure if there's that good understanding why exercise promotes growth.
Yeah, so for people joining via video or teleconference, basically the question here was, to what extent do we understand the mechanism of bone growth and whether an increase in blood flow that might deliver hormones or whatever could explain part of the basis for bone growth? I mean, the very short answer to your question is that I talk about this from a non-NIRS perspective quite a bit in some of the courses that I teach. And we know that bone cells are very mechanosensitive. So even the small amount of loading that you get from the weight bearing associated with just simple walking is enough to physically stress those cells, which reside in little lakes of fluid, and that fluid is squished out through canals to the margins of the bone. And as that fluid flow happens, the cells release growth signals to each other progressively. So that's part of how those bones sense that they're supposed to grow and divide and produce new bone either in width in adults or in height in young people. But we don't know about the blood flow part at all, and that's part of the reason why I want to look at this. Can we promote increased blood flow through exercise, and does that contribute to restoring bone mineral density during osteoporosis, for example? But we don't know. So this is why Colton was really excited to try to do a first ever fundamental characterization of bone optical properties, because there's no better way, we think, to actually look at real-time metabolism in bone during exercise. Any questions from the online folks? No? Okay. Assuming there are some still. Martha.
When you talk about it being NIRS being non-invasive, what you talked about in terms of muscle and bone, what about brain? Is there any difference? Because you've got to go through the skull and then look at the brain. So how, I don't quite get that.
So near-infrared light does pass through bone. We know that. The attenuation of the signal, meaning the degree to which the magnitude of the signal is decreased by bone, is fairly large. So I remember Ken Perkichin, for example, really pressing me on this at one point. And the best evidence that we have from the literature is that the penetration we get below the surface of the interior surface of the skull with near-infrared light is somewhere in the range of 2 to 5 millimeters. So it's not a lot, but it's certainly enough to access the capillaries that feed through the meninges that line the brain, which is what we're looking for. So, I mean, this is the disadvantage compared to something like, say, MRI. We can't look at deep structures like the thalamus or the basal ganglia or whatever. Like, we couldn't do a study related to Parkinson's disease very effectively because we cannot look at the basal ganglia. But we can look at the superficial structures, and NIRS is really good for doing what Cavell is doing, which is looking at the prefrontal cortex, so executive function. So it is non-invasive. Now, there's a caveat, which is that we now have, I'm forgetting the term. They're using scientifically in research settings light wavelengths that will activate certain genes. Anybody know what I'm talking about?
Sort of. There's some that activate.
Yeah.
Like,
I forget. There's opto something. I mean,
Even readers.
okay. Yeah, so I mean, I'm not suggesting to any extent that the neural tissue that's being bombarded with near-infrared photons is having its genetic expression modified. But we already know from some more recent studies that by sending NIRS into the brain, we can measure things totally other than hemoglobin. Like we can measure indirectly changes in the morphology of neurons in real time, non-invasively through the skull under certain experimental manipulations. So there's other stuff going on and we don't fully know. So people often say, oh, well, this near-infrared spectroscopy, it must have side effects. Well, all I can say is that it's been used since 1976 and there's nothing documented yet. Yeah. It's so complicated. We subtract oxygenated hemoglobin from deoxygenated hemoglobin. That's it. And the crazy thing about some of these indices is people say, that can't be real. You're not measuring what you think you're measuring. But the way that we know is that some crazy Scandinavian scientists have done incredibly invasive stuff where they've catheterized carotid arteries and jugular veins and even the aortic arch in some cases to directly measure blood gases at the same time as they do the NIRS measurement. And they've confirmed that these very simple indices are direct measurements or at least very reliable estimates of that parameter.
Very good. So Luke, as you know, I always like the connections between things, and I appreciated your opening by describing yourself as a generalist. And I think you gave us some really, this sort of whole idea of bone, muscle, brain interaction is fascinating. And some of most of your examples were giving kind of couples,
yeah.
of interaction. And I just wonder if sort of if you step back to the biggest level of your findings, whether you've seen any of those sort of connections across all three that you're curious about. It may not be that you've done experiments, but that you're interested in seeing how the three-way connection may be underway.
So I know, yeah, I mean, we're scheduled till 2:30 and I don't want to go for too long, but I'll tell you the deeper story about why I chose that title, which I think answers your question. As a PhD student, I started out looking specifically at neuronal property. And I was really interested at that time in how do neurons change after central nervous system injury. As things went on, it became clear that I was going to have to refocus on muscle. But what that meant in terms of my doctoral studies is that I became really passionate about how changes in one system could lead to changes in the other. And it's not necessarily unidirectional. We know that changes in muscle can lead to changes in the nervous system in some cases. I think I was in the fourth year of my PhD when I was at a Rick Hansen Institute event where I was volunteering, and I met a young woman who was a Paralympic skier. And we had a big conversation about how her sport training had benefited her life. And the next year I saw her again, and it was clear that things had not gone so well for her in the last year. And the reason turned out to be that she had been in competition and suffered very severe fractures to both femurs. And I got very curious about this and I started looking into the literature. And at the time I was studying something called spasticity, which is involuntary muscle contractions that result from certain types of neuromuscular diseases and nervous system injury. So, I mean, the classic example for those who aren't familiar is that you do the tendon tap reflex at the knee and you get that muscle contraction in response to the tendon tap. So that's a reflex contraction. And we have descending inhibition of those reflexes from the brain in real time. If we have a spinal cord injury, there's no ability to provide that descending inhibition. And so what will happen is you'll get the reflex contraction followed by a swing back, which will lead to a reverberating contraction swing cycle. So that's the most simple explanation of spasticity. And the more I talked to her, the more I discovered that because of her sport, she was on very, very heavy doses of anti-spastic medication. And those anti-spastic medications were having a fairly severe impact on her health. And one of the things I ran across was studies where they found that baclofen For example, as one antispastic drug being used to treat muscle spasticity led to more osteoporosis. Because without those involuntary reflex contractions, the bones weren't being loaded. And so that got me thinking, and this led to the introduction I wrote for my doctoral thesis, being very heavily focusing on how exercise interventions were much more beneficial in certain situations than pharmacological interventions. So to answer your question, how do the three interact? Well, I'm not going to speak to the brain, but I will speak to the neuronal tissue in the spinal cord. I spent 2016-17 on sabbatical in Vancouver. I encountered a physical rehabilitation specialist there who was really interested in spasticity. And so what we started doing is using NIRS to measure changes in muscle associated with spasticity. And one of my interests longer term is trying to get a better sense of this, hopefully as an outcome of Colton's work. Because the interaction is the following. If you give certain antispastic drugs, and specifically what I'm going to talk about is botulinum toxin, that toxin induces paralysis at the neuromuscular junction. Unfortunately, if you inject intramuscularly, that toxin travels up the axon to the cell body in the spinal cord. It translocates to an adjacent cell body in the spinal cord and can then travel down that axon. And so in cases where you have incomplete spinal cord injuries, where part of the cord is severed but the other part's not, what you can get, for example, is somebody having an injection to the lower extremity for spasticity. The toxin, the neurotoxin, which eliminates the spasticity, travels up, translocates, then ends up paralyzing the neuromuscular junction in an arm muscle, thereby compromising wheelchair function because the person can't roll anymore. Or if they're getting injections in their arm muscles, travels to the cell, travels down another one, you end up with compromise in respiratory function, which you obviously don't want. So here we have a situation where we're providing a therapy that's meant to treat a muscle disorder. Indirectly through the nervous system, it ends up affecting other muscles. And as a result of that, you're also going to compromise your bone structure in the long term. So I try to think sort of integratively about how can we improve the way in which we deliver a treatment like that so that all three tissues remain healthy. And then, of course, we end up talking about things like the skeletal muscle pump that assists cardiovascular function and the fact that the primary secondary condition we get in people with spinal cord injury is cardiovascular. So then we're looking beyond those three systems. That good?
Yeah, that was great.
Okay. Chelsea.
Any other questions? All right, I think that's it then. Thank you very much. Thank you.